I am Celine Hilda Mary, Assistant Professor in St.Joseph College of Arts and Science, Cuddalore. I am going to present about the blood grouping and RH factor. The ABO blood group system was discovered by the scientist Carl Landsteiner in 1901. He discovered the RH factor in 1930 along with Alexander S. Weiner. For that, he got the Nobel Prize in Physiology in 1930. What are the different blood groups? The differences in human blood are due to the presence or absence of certain protein molecules called antigens and antibodies. The antigens are located on the surface of the RBCs and the antibodies are in the blood plasma. Individual has different types and combination of these molecules. The blood group you belong to depends on what you have inherited from your parents. There are more than 20 genetically determined blood group systems known today. The ABO and RECIS, that is RH system, are the most important ones used for the blood transfusions. Not all blood groups are compatible with each other. Mixing incompatible blood groups leads to blood clumping or occultination, which is dangerous for individuals. Landsteiner law. If an antigen or occultinogen is present on the red blood cells of an individual, the corresponding antibody or occultinine will be absent in the plasma. If an antigen or occultinogen is absent on the red cell membrane of an individual, the corresponding antibody or occultinine will be present in the plasma. That is the Landsteiner law about the blood grouping. Blood group systems. There are different blood groups systems are present. Among these, there are two major blood groups and different minor blood groups are present. The major blood groups are belongs to ABO and RECIS, that is RH blood system. The minor groups are included M1, LIP, Lewis, Duffy, Kidd, Hell, Lutheran. These are the different types of the minor blood group systems. First, we will say about the classical ABO blood grouping system, which is the most important in assuring a safe blood transfusion, which is based on the presence or absence of A and B antigen on red cell membrane. There are four blood groups according to this system. They are A, B, AB and O. This slide shows about the pictorial representation of the antigen and different types of the antigen present in the different blood groups. For example, in the blood group A, the red cell membrane has the A antigen and its surface, and the B antibody is present in the plasma. Likewise, if the person belongs to B blood group, means they are having the B antigen on their surface of the red cell membrane, and they are having the opposite form of the antibody, that is A antibody is present in the plasma. Likewise, if the person having the AB blood group, both the antigens A and B are present on the surface of the RBC cells, but there is no antibody is either A or B antibody is present in the plasma. But in the opposite form is present in the O blood group. In the O blood group, neither A antigen nor B antigen is present on the surface of the RBC, but they are having both the antibody A as well as the antibody B. This slide also shows the presence of the antigen on the RBC cells. In type A antigen is present on the surface of the RBC, and opposite form of the antibody, that is antibody B, is present in the plasma. Likewise, in B blood group, B antigen is present on the surface. The opposite form of the antibody A is present on the plasma. In the AB blood group, both A and B antigens are present on the surface, but neither antibodies are present in the plasma. But in O blood group, no antigens are present in the cell, the RBC cell, but both antibody A and anti B is present in the plasma. So due to the presence of the absence of these both the antigen in the surface, the O blood group person can donate the blood to all the blood groups because there is no antigen is present in the RBC in order to react with the corresponding antibodies. So due to this reason, the O blood group persons are called as the universal donors, but at the same time AB blood group is known as the universal acceptor because there is no antibody is present in the plasma, so they can accept the blood from all the blood group like AB and as well as the O blood group. So they are known as the universal acceptors, but they cannot donate the blood to anybody because of the presence of both the antigen A as well as the B on the surface. So they can donate the blood only to the AB blood group persons only. This slide also shows the different type presence of the antigen as well as the corresponding antibody on the blood plasma in different blood groups of the ABO blood group system. Why do individuals produce antibodies to antigens they do not have? The A and B antigens are also produced by some other plants and microorganisms. Thus, individuals who do not recognize one or more of these antigens as self will produce antibodies against the plant or microbial antigens. These antibodies will also react with human antigens of the same kind, whether introduced via a blood transfusion or a tissue graft. This slide shows the determination of the blood groups. How do we determine the different blood groups? We can determine the different types of the blood groups by using the anti-A as well as the anti-B and anti-D substance. They are having the antibody in its solution, so they are known as the anti-A, anti-B and anti-D. The anti-D helps to determine the RH blood group. In case of the anti-A, for the determination, first we have to take 3 drops of the blood in a clean slide. In the first drop we have to add the anti-D for the determination of RH factor, and second drop, which is the anti-A, is added, and third drop anti-B is added. If the person belongs to A blood group, means the after the addition of the anti-A, that is in the second drop, there is a occultination is occurs. This occultination formation indicates that person is belongs to the A blood group, and if the person is belongs to B blood group, means there is a occultination is occur in the after the addition of the anti-B, that is in the third drop. For in the AB blood group person, both A and B, there is a occultination is occurs after the addition of anti-A and the anti-B, so that indicates the presence of AB blood group. But in the opposite form is occurs in the O blood group, because if there is no antigen is present in the O blood system, so there is, after the addition of anti-A or anti-B, there is no occultination is occurs in the O blood group system, that indicates the presence of the O blood group. In this way we can determine the different types of the blood groups. Next we will see about the genetics and inheritance of ABO system. The ABO gene locus is located on the chromosome number 9. A and B blood groups are dominant over the O blood group. A and B blood group genes are co-dominant. Each person has 2 copies of genes coding for their ABO blood group. One comes from the maternal and one comes from the paternal origin. Next, we'll move on to the RH factor. The rhesus factor gets its name from experiments conducted in 1937 by scientists Carl Landsteiner and Alexander S. Wiener. Their experiments involved the rabbits which when injected with the rhesus monkey's red blood cells produced an antigen that is present in the red blood cells of many humans, that is known as the RH factor, which is first discovered from the rhesus monkey, so which is named as the RH factor. Next, RH blood grouping system, which was first demonstrated in rhesus monkey. These blood groups are classified as RH positive and RH negative. In the RH factor, RH positive and RH negative usually refers specifically to the presence or absence of the antigen D. There are two alleles or genetic variants of this antigen, capital D and small d. A person who is Rh negative has two recessive traits, D, small d and small d. Anyone who has at least one capital D or two capital D or one capital D and one small d is belongs to the Rh positive. RH type and pregnancy. A person's RH type is generally most relevant with respect to pregnancies. If the pregnant woman and her husband are RH negative, there is no reason to worry about RH incompatibility. But if a pregnant woman carries the RH positive blood group and the father has the RH negative blood group, that leads to the RH incompatibility after the first delivery, that is during the second delivery, which leads during the formation of the second fetus, which leads to the complication, that is destruction of the fetus, that is leads to the hemolytic disease. RH incompatibility is a condition which it develops when there is a difference in RH blood type between that of the pregnant mother if she belongs to RH negative and that of the fetus RH positive. Usually, placenta acts as barrier to fetal blood entering maternal circulation. However, sometimes during pregnancy or birth, feto-maternal hemorrhage can occur. The woman's immune system reacts by producing anti-D antibodies that causes sensitization. Fetal cells enter maternal circulation through the break in placental barrier, example at placental separation. In the next stage, maternal production of rhesus antibodies following introduction of the positive blood. After the first delivery, in the RH positive blood substances antigens may enter into the maternal blood and which involves the production of the antibody against the RH positive antigens. In a subsequent pregnancy, maternal rhesus antibodies cross the placenta, resulting in a hemolytic disease of newborn. That is, in the hemolytic disease of fetus and newborn caused by RH isoimmunization can occur during the first pregnancy, but usually sensitization during the first pregnancy or birth leads to extensive destruction of fetal RBC during subsequent pregnancies. These are the conditions which affect the first pregnancy. They are miscarriage, abortion, feto-maternal hemorrhage. Next, we will move on to the sign and symptoms of RH incompatibility. RH incompatibility can cause symptoms ranging from very mild to fatal. Mild test form RH incompatibility, which leads to hemolysis and jaundice. In case of hemolysis, destruction of our blood cells with the release of free hemoglobin into the infant circulation. In the jaundice, hemoglobin is converted into bilirubin, which causes an infant to become yellow. That leads to they are occurs due to the RH incompatibility. This is the picture of the fetal hemolysis. Fetal hemolysis. This is the picture of neonatal jaundice. Next, we will move on to the detection of RH incompatibility. There are no any physical symptoms can seen in rich RH incompatibility. If the woman just found out she is pregnant, she should undergo blood type test. This test determines her blood type and RH factor. Blood test that will determine whether she is RH positive or RH negative. This is the phototherapy of the neonatal jaundice. Prevention: by using special immune globulins called ROGAM at 28 weeks of pregnancy, we can minimize the hemolytic disease. If the baby is born RH positive, another dose is administered within 30 to 72 hours after delivery. This will prevent her body from creating a future antibodies that will cause harm during pregnancy. Transfusion incompatibility, which means wrong transfusion can cause occultation of blood in the recipient. Henceforth, transfusion of blood along with identification of ABO blood group, it is necessary to test compatibility of RH factor also. Summary: RH negative blood mother, if the mother carries the RH negative blood, the RH the father may be RH positive, so the baby sometimes may have the RH positive blood group. So in the RH positive blood group may enters into the maternal blood circulation and act as an antigen and produce the antibodies against the RH positive. This antibody persists in the mother's womb, but which after this first delivery, which is persist in the mother's womb, but when the second baby is formed, the same antibody enters into the fetus through the placenta and destroy the fetus. That is known as the erythroblastosis fetalis. It is a hemolytic disease. Next, what are the clinical application of blood grouping? The detection of blood group helps for the transfusion of the blood, which prevents the hemolytic, helps to prevent the hemolytic disease. In paternity disturbs in medico-legal cases, in knowing susceptibility to disease. Group O duodenal cancer, Group A carcinoma of stomach, pancreas and salivary glands. These disease conditions can be diagnosed with the help of the detection of the different types of the blood grouping. Next one is the Bombay blood group. Bombay blood group is the one which is discovered in the Bombay. That means here H antigen or H substance is absent. The deficiency of the H antigen was discovered in Bombay by Dr. Y.M. Menin. So this blood group is known as the Bombay blood group, which is present in about 0.0004% only. That's all about the classification of the different types of blood group and RH factor. Thank you.